Hello, everyone, and thank you for joining us today. My name is Sharia Kennedy, and I'm an account representative at Omega Bio-tek. It is my pleasure to moderate our webinar entitled Extraction Strategies for Comprehensive DNA and RNA Analysis from the Same FFPE Sample. First, I will take a few minutes to introduce our speaker and a little bit more information about Omega Bio-tek. Immediately following the presentation, we will host a brief Q&A, so please feel free to submit any questions you have through our chat. Again, my name is Sharia, and I will be your moderator for today's webinar, which will be presented by Dr. Kiran Durvasula, who is a product manager at Omega Bio-tek. She currently leads efforts to develop and deliver novel nucleic acid isolation products for various research and diagnostic fields. Dr. Durvasula obtained her PhD in chemical engineering from the University of Florida, Gainesville, and went on to receive postdoctoral training at the Georgia Institute of Technology. From there, Dr. Durvasula served as a biological scientist at Open Cell Technologies Incorporated, developing an innovative intracellular delivery and transfection device before joining the team at Omega Bio-tek. She has authored several peer-reviewed journal papers and is passionate about translating fundamental research platforms into practical technologies. For those of you who aren't familiar with us, Omega Bio-tek is a manufacturer and distributor of nucleic acid purification products based in Atlanta, Georgia. Since 1998, we have been dedicated to delivering an exceptional customer experience by providing top-notch support and innovative purification solutions to labs around the world. Our diverse product portfolio offers 3 different purification formats, which include your traditional spin columns for manual processing, 96-well filter plates for medium throughputs, and magnetic beads for automated workflows. Omega has developed robust DNA and RNA purification kits for a wide variety of applications, including, but not limited to, basic, translational, and clinical research, molecular diagnostics, agricultural, and environmental applications. When working with Omega Bio-tek, you can expect to receive quality control products that deliver robust performance and ease of use. By offering multiple purification formats and product sizes, such as individual components and custom kits, our product line is suitable for a wide range of throughputs. The Omega team is always available to answer your questions and offer tailored recommendations for your specific application. If you work with a magnetic processor or liquid handler, our app scientists can provide consultation, scripting, and installation services to get the automated workflows up and running to your satisfaction. In summary, Omega's high-quality products, flexible ordering solutions, and exceptional customer support can help you achieve the best economical value for your consumable costs. Now, I would like to hand it over to Dr. Kiran Durvasula to present today's webinar.
Thank you, Sharia. So before I move on to the crux of this webinar, talking about the extraction solution from Omega Bio-tek from FFPE samples, I would like to take a moment or two to talk about the FFPE sample in general and the challenges of working with these FFPE samples. So FFPE samples are most common sources of archive material for tissues. As you all know, FFPE stands for formalin-fixed paraffin-embedded. And this type of preservation ensures long-term preservation of tissue with intact structures. FFPE samples are invaluable biospecimens, aiding the fields of cancer genomics, pathological diagnosis, immunological research, biomarker discovery, etc. However, working with these samples involves a lot of challenges, especially in terms of getting high quality DNA and RNA from these samples. The reasons are myriad. For example, it can be limited starting material that's available, and often these samples are low yielding. And depending on the tissue type, age, and fixation procedure, the DNA and RNA are often fragmented and of poor quality, and the nucleic acids and proteins are often cross-linked. And if you look at the conventional nucleic acid extraction strategies used, they often involve use of toxic chemicals such as xylene and other organic solvents for deparaffinization. However, unlocking information from this FFPE sample is very important to fully realize their downstream potential. Conventional methods, as I mentioned before, require the researcher to either pick DNA or RNA to extract from the sample, or what would happen in that case is like the researcher would end up losing the other nucleic acid. Or the other conventional technique that's typically used involves splitting the sample. So essentially, the researcher is extracting DNA from one part and RNA from the other part. These conventional methods have major drawbacks. One, by splitting the sample, you are already splitting a low yielding sample and often results in even lower yields, which are not really enough to do any sort of meaningful downstream. And two, the researcher is not able to get a comprehensive nucleic acid profile from the sample because either one, they just have to pick one over the other, or two, even in the case of splitting the sample, the cell source can be different from one part to another. So, to answer this need, Omega Bio-tek has come up with an innovative nucleic acid extraction solution with which we're able to sequentially isolate DNA and RNA in two separate eluates from the same FFPE sample. The big advantage with our kit is there's no need to split the sample. We're able to extract DNA and RNA from the same sample, essentially giving the researcher that ability to get comprehensive nucleic acid profile from the sample. This kit is magnetic bead-based, so the full protocol after the deparaffinization step can be automated. We use non-toxic mineral oil instead of xylene for deparaffinization for one, to reduce the hazardous chemicals being used in the lab, and two, give the researcher that ability to do non-toxic mineral oil instead of xylene. Our kit also uses a specialized buffer system to partially reverse the formaldehyde-induced crosslinking in the DNA and RNA. So I want to briefly go over the protocol with you all. So it starts with the deparaffinization step in the presence of mineral oil. So after a brief incubation step, the sample is lysed in the presence of pyranase K for the tissue digestion. And the lysate is then heated to one, inactivate the pyranase K, and two, to promote the decrosslinking of nucleic acids. So after that step, we add binding buffer and magnetic beads to bind DNA to these magnetic beads. And the RNA containing supernatant is actually saved. And we add more alcohol and the RNA is bound to a separate set of beads. So like this, we're actually able to separate the DNA and RNA into two different fractions. So both of those fractions containing the DNA and RNA bound beads are washed separately and eluted into two different fractions. So essentially we have a tube containing DNA and a tube containing RNA, and they're ready for downstream analysis. So talking about the performance from this kit, we've used FFPE samples from 3 tissues: breast, lung, and colorectal. And we've used our MagBind FFPE DNA and RNA kit and compared it to two leading manufacturers' kits. One is column-based from company Q, and the other being the magnetic bead-based kit from company T. So post-extraction, we put DNA and RNA through a different variety of downstream analysis. One being nanodrop, where we looked at the yield and the quality of the DNA and RNA extracted by looking at the absorbance ratios of 260-280 and 260-230. So we also quantified the DNA and RNA using Promega's fluorescence-based dsDNA and RNA system to specifically quantify DNA and RNA without interference from each other. So the extracted DNA and RNA is also amplified using real-time PCR to look at the amplification potential of the extracted DNA and RNA, and also the sensitivity of detection. Last but not the least, we also did next-generation sequencing using Illumina's TrueSight Tumor 170 panel to look at the sequencing results of the DNA and RNA. So this panel actually takes the input of both DNA and RNA. It covers a wide range of tumor genes and variant types. So moving on to the results. I want to draw your attention to the solid color columns, which are our DNA and RNA quantified using nanodrop. And the shaded columns are our DNA and RNA quantification following the QuantiFluor system. So looking at the left graph, if you look at the box that is highlighted, you can see that from the lung and the breast tumor samples, our yields from Omega Bio-tek kit are significantly higher compared to company T and company Q, whereas for the colorectal sample, they're comparable. And looking at the graph on the right of the slide, for the lung tumor samples, the yields following Omega Bio-tek's extraction was significantly higher compared to company T and company Q. Whereas for the breast and colorectal, they're comparable with company T. And for all the tissue samples that were tested, Omega Bio-tek's extraction yielded RNA significantly higher than company Q. Looking at the performance metrics of purity, we looked at the 260-80 ratio as well as the 260-30 ratio. So the solid color here is a 260-80. So if you look at the average 260-80 for DNA and RNA following the Omega Bio-tek's protocol, they're very close to the theoretical 1.8 and 2, which indicates pure DNA and RNA. For the company T and company Q as well, the ratios were very close to 1.8 and 2. But however, if you look at the 260-30 ratio, it tells a different story. The Omega Bio-tek's, irrespective of DNA and RNA that was purified, the ratios are greater than 1.4, whereas with the other two kits, they were significantly lower than the Omega Bio-tek's, which suggests that the nucleic acids extracted using the Omega Bio-tek's kit were of superior quality than those extracted using the other two kits. So I want to briefly go over the qPCR analysis that was done on the extracted DNA and RNA. So if you look at I just chose to highlight the colorectal tissue samples just for the sake of simplicity here, because it can get very busy. So if you look at the colorectal tissue samples, the Omega Bio-tek's protocol actually amplified at the lower CT, suggesting better amplification potential and higher sensitivity compared to the other two extraction kits. So other important performance metrics when we talk about FFPE samples is like how it compares with the fresh frozen, because at the end of the day, you want your FFPE sample to be a true representation of your fresh frozen sample. So on this slide, let me draw your attention to the last column here, which is the delta CT. So that is actually the cycle number relative to that of fresh frozen. So the delta CT, lower delta CT basically indicates that the quality is higher and it's a better representation of the actual tissue sample prior to FFPE processing. So with Omega Bio-tek kit, we got a delta CT of 3.1 compared to 4.06 from company T and 5.32 with company Q. So what it tells us is basically with the Omega Bio-tek's processing, we're able to get higher nucleic acid quality, and the result is more closer to that of fresh frozen, indicating a better representation of the fresh frozen sample. Just a note here, the NGS panel that we chose, Illumina recommends a delta CT of less than 5 for optimal performance of the TrueSight panel. And looking at the delta CT, the company T and Omega Bio-tek both actually satisfy this requirement, whereas the company Q fails to satisfy this criterion. So DB200 is a very interesting metric. This actually accurately assesses the quality of the RNA derived from FFPE tissues. So DB200, what it tells us, it's basically gives us the percentage of fragments greater than 200 nucleotides. And DB200 is an incredible tool for researchers because it allows them to reliably quantify the degraded RNA in terms of size, and gives them an opportunity to effectively parse tissue samples that are suitable for NGS from unsuitable samples, conserving both time and money. Again, I want to draw your attention to the colorectal sample just for the sake of simplicity. As you can see, we have better DB200 with Omega Bio-tek compared to company T or company Q, and you see a similar story across the breast and lung tissue samples as well. What it tells us is, with Omega Bio-tek's extraction, we're able to extract superior quality RNA compared to the other two kits. Again, a note here, the Illumina's NGS panel that we chose, the DB200 requirement is 20%. So in that sense, irrespective of the extraction methodology used, the RNA quality RNA quality irrespective of the extraction methodology satisfies that requirement. Looking at the sequencing metrics here, again, I'm drawing your attention to the colorectal tissue sample. The quality metrics compared to the fresh frozen Omega Bio-tek's is closest, suggesting it's a more accurate representation of the non-FFP sample. And looking at the median insert size and exon basis of 250x coverage, they're all very comparable irrespective of the extraction methodology. And all of these extraction kits satisfy the quality threshold of 79 base pairs for median insert size, which was set forth by Illumina's TrueSight 170 panel. So looking at a small variant calling, same mutations were called irrespective of the extraction kit used. However, if you look at the allelic frequencies and the depth of coverage, especially for the TP53 mutation, the Omega Bio-tek seems to be most in concordance with the fresh frozen, allowing for the same conclusions to be drawn. Again, I'm just showing the colorectal tumor tissue sample data here. We do have data with the FFP breast and lung tissue samples, but that data is not shown here. So coming to the overall conclusions, the Omega Bio-tek's MACBA and FFPE DNA and RNA kit can extract both DNA and RNA from the same tissue sample without sample splitting, and it exhibited superior performance when compared to kits from company T and company Q in terms of yield, quality, as well as qPCR functionality. As I've shown the results in the previous slides, they indicate very efficient separation of DNA and RNA into separate eluates following the Omega Bio-tek's kit. The Omega Bio-tek's kit follows a magnetic bead-based approach, making it automation and high throughput friendly. And that's a distinct advantage over the company Q's kit, which is just column-based. Since I mentioned a lot about automation, I just want to briefly talk about the automation capabilities of this kit. So we do have a very experienced field application scientist team who are able to provide scripts with our kits as well as help you in scripting. And on this slide, I have listed all the liquid handlers and magnetic processors that we support. So if you are using one of these magnetic processors or liquid handlers, we are able to help you in scripting of this kit or any of our other kits as well. With that, I thank you all for your attention and pass it over to our moderator. Thank you.
Thank you, Kiran. Now, we'd like to take some time to answer some questions from the audience. Let's see here. First question. How old were the samples used in these analyses? And have you tried this kit in other tissues or species?
Thanks, Sharia. So the samples are relatively new. I do not have the exact age right now, but these are not like super old samples or anything. So we did try this kit with other tissues, like liver. Liver is something that we've tried. I do not have the data right now to present. As far as talking about the other species, we have not personally tested in other species, but we have customers who do use this kit in like, varied applications. They've used it for like different aged tissues, as well as like in, mouse liver tissues and stuff like that.
Great.
Yeah.
Okay. So next question. Can this kit be used for next generation sequencing?
Yes, definitely. The kit can be used for next generation sequencing. However, so whatever panel that you choose, my recommendation would be to, check the quality metrics needed for that. So once you do the DNA and RNA extraction, and once you quantify your DNA and the quality of DNA and RNA, do check with the metrics needed for that particular NGS panel. And we have just presented sequencing data in this webinar, so definitely be used for sequencing.
Okay, I have another question for you, Kieran. My FFPE sample is overfixated. Can it still be used?
Overfixation, I mean, it definitely can be used, but however, problem with overfixation is like, the DNA gets heavily cross-linked. So the reversal of that cross-linking often gets difficult because of overfixation, and it does cause like poorer quality nucleic acids downstream. So we do have few recommendations, like we can extend the cross-linking time. As I presented the protocol, we typically do 90 degrees C for 1 hour. We can definitely extend it by half an hour more, and that could remedy that situation. However, if we extend it for a longer time, we do run the risk of degrading the RNA. So it's a fine balance there. Yeah.
Okay, so next question is, how would I modify the protocol for an automated workflow? I'll go ahead and take this one. So
Okay.
Our FAS team specializes in customizing our workflows for automation platforms. They're mentioned at the end of the presentation, such as Hamilton Star, TCAM Fluent, Thermo Kingfisher, and other workstations. We also offer consultation, scripting, and installation services, which go to help you implement a tailored workflow for your specific needs, because everyone's working with something a little different. We like to work closely with you all on developing something that works best for you. So if you just need a script, or if you have some questions, or just need any further support whatsoever with your automated workflow, I would recommend going to our website and submitting an automation support ticket, and that will connect you with your local app site. The next question. Can the proteinase K digestion be carried out overnight? And how will it affect the RNA yield and quality?
Yeah, that's a very good question. So definitely the proteinase K digestion can be carried out overnight instead of, I think the protocol calls for 4 hours. In fact, a lot of our customers do prefer to do it overnight, because you can set up your digestion the day before, go home, come back the next day, and just jump into purification part. So it does ease the workflow, but better by setting it up like that. We did validate that at our facility, at our research facility. So when we did incubation, proteinase K incubation for 4 hours versus overnight, we saw comparable DNA and RNA yield and quality. So I don't believe it'll affect the RNA yield and quality. So go ahead and do it overnight.
Okay, looking at the time, looks like we have time for at least a couple more questions.
Okay.
So if I only need FFPE RNA, would this be the right kit to use? So that's a good question. So if you're interested in RNA only, we would actually recommend our MagBind FFPE RNA kit. And similarly, if you're only interested in DNA, we have a MagBind FFPE DNA kit as well. And all those options and more information can be found on our website. Also, one last question for you, Karen. Can we use the same kit for extracting DNA and RNA from fresh frozen tissue?
Very good question. So we are in the process of developing a similar sequential DNA and RNA isolation kit from tissues and cells. So that would be something that'll be coming out like next year. So do check back with us. But in the meanwhile, if you do have to extract DNA and RNA from fresh frozen tissue, we may be able to provide a modified protocol using this kit. It's not going to be the most ideal protocol, but we can provide a modification on the existing protocol. So do connect with our product support team, and they can help you with that.
Awesome. Well, that concludes our presentation for today. I'd like to thank Kieran and everyone that joined us. If we didn't have time to answer your question, an Omega representative will be following up with you in the days to come, so look out for that. Please visit our website or contact us using the information provided on the screen below if you would like a product sample, have any questions, or would like to request additional support for your DNA and RNA purification workflow. Thank you again for joining us and have a wonderful day.
Yeah, thank you, everybody. Bye-bye.
Thank you.